Hello, this is Goal 2, Module 3, the building blocks and macromolecules. In this module, the third one for Goal 2, will enable you to distinguish between building blocks and macromolecules, between decomposition, synthesis, and exchange reactions, and describe the role of enzymes in chemical reactions. Building blocks make up larger substances called macromolecules. The four macromolecules are all made of many monomers, which are repeating units of the same thing. For example, a carbohydrate is a macromolecule or a polymer made up of monomers, usually monosaccharides like glucose. Simple sugars or monosaccharides make up carbohydrates or polysaccharides. Fatty acids make up lipids or fats. Amino acids make up proteins, and nucleotides make up nucleic acids. Just memorize this slide. You will be using these over and over and over. Building blocks are glucose, fatty acids, amino acids, and nucleotides. And macromolecules are the polysaccharides or carbohydrates, the lipids, the proteins, and the nucleic acids. The elements in all the building blocks spell CHOMPS, C-H-O-N-P-S, and include carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. All of the atoms in these molecules are covalently bonded, and therefore when we make them, it takes a little bit of energy, and when we break them, we get a little bit of energy. In general, we eat the macromolecules, and our digestive system breaks them down into building blocks, so our cells can eat the building blocks. Macromolecules are too big to get into our cells, so we need to break them down into the building blocks in order for our cells to be able to use them. Although the definition of organic molecule in chemistry is somewhat arbitrary, here we will consider molecules that contain both carbon and hydrogen to be organic. Those molecules that don't have both will be considered inorganic. It's not that either one is more or less important, but the building blocks are referred to as organic molecules in most textbooks, and molecules such as salts, carbon dioxide, and water are considered inorganic because they don't have both carbon and hydrogen. Weirdly, methane, which is in the diagram here, is considered organic, even though it certainly isn't anything we can use for energy in our bodies. Glucose is the most common building block of carbohydrates. There are other monosaccharides like galactose and fructose, but glucose is by far the most common. The three elements found in carbs are carbon, hydrogen, and oxygen. Glucose can be polymerized into three different types of carbohydrates: starch, cellulose, and glycogen. Starch is what we love to eat, and we digest this very easily. Cellulose, even though it's the same glucose molecules hooked together, are hooked together slightly differently, and we do not digest it very well. We refer to it as fiber. It does add bulk to our diet, helps hold water in the digestive system, and helps keep us regular. We don't get calories from cellulose because we don't really have enzymes to break it down. Plants can make both starch and cellulose, but humans can't make either one. When we store glucose as humans, we polymerize it into glycogen. We can store this in our livers and in our muscles, but only about enough for 12 to 24 hours. The main reason we eat carbs is for energy. Our cells absolutely love glucose. Every cell in our body, and indeed every cell on Earth, can use glucose as a source of energy. Fatty acids are hooked together to form lipids. By definition, when you polymerize them, the most common way we put them together is in the form of triglycerides, three fatty acids hooked to a glycerol molecule. Fatty acids are hydrophobic molecules. Okay? Remember, that means they don't like water. Therefore, will separate from water. Sometimes we even refer to lipids as grease. Triglycerides are the main type of fat in the food we eat, and cholesterol, and adipose tissue is where we store it. Fats contain the elements carbon, hydrogen, and oxygen just like carbs, although there's much less oxygen in a fat. Besides triglycerides, the main form of fat we eat, there are also phospholipids and cholesterol, both a part of our cell membranes. Cholesterol is also used to make steroid hormones in the human body, so you need some cholesterol. If you don't eat it, your body will make it. Fatty acids come in three main types, which you may have heard about or even seen on a food label: saturated, monounsaturated, and polyunsaturated. Saturated means, and you can see in this picture here, that all the bonds between the carbon are single covalent bonds. There are no double bonds in a saturated fatty acid. In an unsaturated fatty acid, there's a double bond. If there's just one, it's a monounsaturated fatty acid, and if there's two or more, it's a polyunsaturated fatty acid. Saturated fats are like butter or the fat on meat. They are solid at room temperature. A monounsaturated fatty acid is softer, like margarine or olive oil, and polyunsaturated fat at room temperature is almost always a liquid, like canola oil or fish oil. Our bodies can make most fatty acids, but there are two you absolutely need to consume in your diet: linoleic acid and linolenic acid. Remember the rule about solid and liquid at room temperature. Saturated fat tends to be solid at room temperature. Monounsaturated is a little softer, and poly is more liquid. Some of the sources of these are listed on this slide here. There's been a lot of health concerns related to fatty acids. Research continues to show that different things about fat. For example, the saturated fats in milk, which they used to think were bad because they were saturated, are now thought to be not so bad. Cholesterol in the diet has now been changed by the FDA as not something to be concerned with because dietary cholesterol doesn't seem to have more than a 10% effect on your blood cholesterol. It's your genetics more than your diet when it comes to cholesterol. Fat does have twice the calories that carbs do, so by weight, you don't want to eat a lot of fat. And if you're trying to lose weight, fat calories are something people tend to try to avoid. Trans fatty acids are considered to be so dangerous now that by 2017, I believe, they have to be taken out of all foods. If there is trans fatty acids in a food, it must be stated on the label. Amino acids, the building blocks of proteins, contain carbon, hydrogen, and oxygen, and also nitrogen, and two of the amino acids contain sulfur. Proteins, while they certainly can provide energy when you break them down just like a carbon of fat, are the functional units of the body and tend not to be broken down unless you need energy. These macromolecules are your enzymes, your receptors, your structural proteins, transporters. They do things for us. They function. We need proteins for more than just energy. While they are made up of covalent or peptide bonds, the hydrogen bonds that cause them to fold are also important, because without proteins folding, they don't work. The unfolding of proteins is called denaturation or denaturing, and it happens when the temperature is above 98.6, a couple degrees above, or when the conditions are acidic or basic. Both of these things will kill us quite rapidly. Remember, even though proteins are made of covalently bonded amino acids, they must fold up. Hydrogen bonds do this, and they must be folded correctly to have functionality. The last building block in macromolecule are the nucleotides, which bind together to form nucleic acids. Nucleic acids, your DNA and RNA, carry genetic information and are made up of the building blocks usually abbreviated A, G, C, T, or U. That's adenine, guanine, cytosine, thymine, and uracil. We'll talk more about these during Code 4. Again, the elements in nucleotides are CH and O, carbon, hydrogen, and oxygen, nitrogen, and phosphorus. The picture on the right is DNA. It is a twisted ladder where the rungs are made up of the bases A's, G's, C's, and T's, and they are hooked together by hydrogen bonds. RNA is only half that ladder, and instead of a T it uses uracil. The sugars are slightly different between DNA and RNA. In DNA it's deoxyribose sugar, and in RNA it's just a ribose sugar. This table summarizes what we've just talked about in the last few slides. Right in the middle of the table are the macromolecules, and the building blocks are to the left of that. The elements in these building blocks are listed in the first column. Some examples are listed in the fourth column, and the function in the body is listed on the far right. Notice that any of the first three can be used as energy. There are three main types of reactions in the human body, most of them involving those building blocks in macromolecules. We break them down, we build them up. When two smaller units are hooked together, like when we hook two glucoses together to form glycogen, that type of reaction is referred to as a synthesis reaction. When we break that glycogen down, that is called a decomposition reaction because you're making something smaller or breaking it into pieces. If you break down a disaccharide into the two monosaccharides, that is decomposition. If you exchange part of one molecule with another, or trade around the atoms, it is referred to as an exchange reaction. You can see that in this slide as well. Synthesis reactions usually require a little bit of energy because you're forming a bond, whereas a decomposition reaction, generally energy is released because you're breaking a bond. So A plus B equals AB is a synthesis reaction. AB broken down to A plus B is a decomposition reaction. Exchange reactions are a little bit less common, but the molecules essentially are trading places. AB plus CD equals AD plus CB. Most reactions are reversible, and in the body, an enzyme that adds A plus B together can also break down AB into A plus B. Looking at these reactions, can you tell which one is which? Okay. The first one is an exchange reaction, the second one is a synthesis reaction, and the third one is a decomposition reaction. The one that's most likely to generate energy is the decomposition because you're breaking a covalent bond. The first reaction did result in some energy. It is considered exothermic because heat is released. The last thing we're going to talk about in this module is an enzyme. Those reactions that we just talked about can happen spontaneously, but it would take forever for that to happen. For example, if you took cookie dough and put it on the counter, it would take a long time for that to turn into a cookie, even if it never rotted and nobody ever snitched it. Enzymes speed up the rate of this reaction. So ovens are a little bit like enzymes. They convert cookie dough into cookies in just 10 minutes. Enzymes do not change themselves. They just convert substrates, the cookie dough in our example, into products, the cookies. As long as certain conditions are met, your oven's plugged in and the gas is on, it will continue to convert cookie dough into cookies as long as you keep putting cookie dough in. The same is true of our enzymes inside our cells, except they're more specific than ovens. Our ovens can bake bread or cookies or pot roast, but an enzyme usually has just one substrate and makes just one product. So you need a different enzyme for each reaction that takes place in your body. On average, cells have thousands of enzymes each one acting like a different kind of oven to convert a substrate into a product. Enzymes are catalysts, so they speed up the rate of reaction. Substrates would not be converted into products fast enough for life to exist as we know it without enzymes. Enzymes are usually proteins, so they fold up in specific shapes based on their hydrogen bonds to fit an exact substrate. It's very important that the correct temperature and pH is maintained so your enzymes work. Can you guess what pH or temperature works best for most of our enzymes? Hopefully you said pH 7.4 and temperature 98.6. Here's another good picture showing the substrate binding the enzyme and being converted to a product. And as soon as that product's released, the enzyme can be used again. Again, the specific shape of the enzyme is based on the hydrogen bonds. So screwing up the temperature or pH will screw up your enzymes, and pretty soon you won't be able to convert any substrates into products. Not only do enzymes need proper temperature and pH, many of them require cofactors to work properly. Cofactors are usually vitamins or minerals. They bind the enzyme, like in this picture up at the top, and change its shape just right so the substrate fits exactly into the active site of the enzyme. Enzymes are always doing something. They're making product. But once a cell has enough of that product and it's not needed anymore, the enzyme is shut off by that product. This is another example of negative feedback. Hopefully you remember that from the homeostasis lecture that we did in Goal 1. Another way to say this is end product inhibition, where the end product shuts off the first enzyme in the pathway so that you don't bother to make extra. Cells are very efficient, unlike people. Again, the resources for this module include Lesson 2, your online textbook, the internet. There's a good link here for enzymes and how they work. Be sure to check that out. Look in your lab packet and don't be afraid to ask questions.